So prokaryotes use the theta model for DNA replication. And DNA replication starts at the origin of replication, which is a sequence of DNA that tells the process where to start. And it begins to melt open, and the theta model is bidirectional replication. So you're going to have a replication fork here and another one here. The replication fork is where single-stranded DNA is unwound from the double-stranded DNA helix. So in order to understand a replication fork, it's important to remember that DNA is anti-parallel. So if you have a 5' to 3' strand, then the complementary strand is going to be 3' to 5'. So helicase binds at the replication fork and it drives into the replication fork, breaking hydrogen bonds as it moves along the DNA. And as this single-stranded DNA is exposed, single-stranded binding proteins bind to the DNA to stabilize it and to prevent double-stranded DNA hairpins from forming. Gyrase is a topoisomerase that is in front of the replication fork to prevent stress and supercoiling from happening, and it does this by nicking a phosphodiester bond in the DNA, and this is a bond that connects sugar molecules and polynucleotides. So in order for new DNA to be added, DNA polymerase 3 requires a free 3' hydroxy to add nucleotides to. So RNA primase lays down short RNA primers, and that's shown here in orange and in all of these, in order for DNA polymerase 3 to be able to add nucleotides. So the strand that's driving into the replication fork is the leading strand, and it's going from 5' to 3' because you always add to the 3' end. And then the lagging strand is the strand that is going away from the replication fork, and this forms Okazaki fragments through discontinuous replication. Every single one of these is an Okazaki fragment. The leading strand only requires one RNA primer, and the lagging strand requires an RNA primer in front of every Okazaki fragment. So DNA polymerase 1 eventually comes along and takes out all of these RNA primers, and then DNA ligase comes and fills in the gaps of these Okazaki fragments. So this is just showing you that the theta model has two replication forks.